Exactly.
Are there any other materialistic processes? I've heard people point to, say, for example, cancer. Sometimes as cancer mutates and changes, novel information arises.
Well, what you have in circumstances like that, and pretty much in all counter examples that have been offered as refutations of the way I made the information case in Darwin's Doubt, which is about already existing life. It is certainly possible, and we have evidence of mutations altering pre-existing protein folds and optimizing them a little bit.
Okay.
But what we don't have is evidence of mutation and selection building novel protein folds. And a protein fold is the sort of fundamental unit of innovation in the biological world. And when Doug Axe did his important work on showing the rarity of functional proteins, what he was really showing was the rarity of protein folds. And proteins have 3 levels of structure, and the tertiary structure is what corresponds to the fold. And that's the basic structure that allows proteins to have this hand-in-glove fit that allows them to catalyze reactions or build molecular machines or process information. So there are several thousand protein folds as fundamental structural units in the living world, and they are incredibly rare among all the different ways there are of arranging the subunits that make them up, called amino acids.
Mm-hmm.
So that's what we dispute mutation and selection can find, is a new protein fold. We're perfectly happy to acknowledge that mutations can sometimes optimize existing folds and give them slight differences. And that's when people point to new information arising by mutation, it's typically a slightly modified or optimized pre-existing protein fold.
Okay. How about in the Return of the God hypothesis? Darwin's Doubt was newer, but clearly there have been some naturalistic attempts to explain the Cambrian explosion. What's one of the leading ones, and maybe a response that you give?
Well, that's actually one of the big ones right there. There were, because at the fundamental level, what I argued was that new protein folds are beyond the reach of the mutation selection mechanism. Axe found that in one particular protein that he was studying, beta-lactamase, that for every sequence of amino acids that did form a stable, what he called a function-ready fold, a fold capable of performing biological functions, for everyone that did, there were 10 to the 77th power amino acid strings or sequences that did not. And so he had this very powerful mathematical argument against the plausibility of randomly searching a space of possibilities that large in the given full history of life on Earth. And so there have been counter arguments to that, one made by some of the theistic evolutionists, in fact, Dennis Venema and Deborah Harzma. And I responded directly to that. They claimed, for example, that a protein called Nylonase, which is a protein that can gobble up synthetic nylon, which has only been around for about 40 years, showed that Axe was wrong, because a few simple mutations, and voila, you had a new protein that was functional, and they sort of implied that it was a fold. But when you went back and looked at the original Japanese research group that studied the Nylonase protein, it turned out that it was a couple of simple point mutations within an existing fold structure. They did not alter the fold, and the Japanese researchers were totally explicit about that.